Hey everyone. So as I mentioned, my name is Daniel Burkhardt, and today I'm really excited to have the opportunity to talk to you all about some of the work that I've been helping to organize in an effort called Open Problems in Single-Cell Analysis. As a little bit of background, I started this project when I was a PhD student and submitted to Kurswami's lab in collaboration with some other folks in the group, some great scientists at CZI Biohub and Helmholtz. And it's been a really, excellent community project I've been able to take part in. As I also mentioned, these days I work at Celerity, which is a Cambridge-based biotechnology startup. I'm working with a bunch of folks with experts in single-cell biology, systems biology, machine learning, and we're working on trying to identify novel therapeutics by focusing at the level of individual cells as opposed to particular molecular pathway. And what's really wonderful is that in this role at Celerity, I'm also able to continue to work on these kinds of open science and community projects that I started doing in my PhD. And, it's been really wonderful to get to continue to do some of that work. So this project, Open Problems in Single-Cell Analysis, really started in 2019 when I was first trying to understand how we can build and identify best practices in single-cell analysis. I came across this paper from Malte Lueken and Fabian Theis. And in Smith's lab at Yale, we started to look through and understand the environment of benchmarks that people were using for establishing what best practices were for doing filtering and normalization and clustering and visualization. And what we noticed is that there are dozens and dozens of independent benchmarks. Each of these typically identify a particular metric or data set that they're interested in, and then oftentimes, that is sort of the end of it. And these best practices then get replaced by new benchmarks using new metrics on new data sets. And so there wasn't really this kind of consistency that we observed. And it's really interesting to think about if there's a different way to sort of organize these longitudinal, continuously updated benchmarks. Around the same time, I also went along with a bunch of other folks in the lab to a conference called NeurIPS, which stands for Neural Information Processing System in Vancouver. So this is a picture from December 2019 when conferences were in person at a workshop called Learning Meaningful Representations of Life. This was one of the only life sciences related events at NeurIPS, which is the largest machine learning and artificial intelligence conference in the world. There's about 15,000 people that attend. There are about 800 people who filtered through this workshop. It was overcrowded. There's a bunch of folks you can't see out in the hallway. And it was a really exciting opportunity to see this huge amount of excitement from the machine learning community to understand what were the existing problems in biology. But as I started to talk to folks after the workshop, I realized that there's a common problem where individuals who had a background in computer science and machine learning were really interested in getting involved and working in the life sciences, but didn't really know how to get started with tackling those kinds of problems. And so I ended up going to a coffee shop in Vancouver with Joshua Batson from CZI and Scott Gigante from my lab, and then had some further conversations with Smitha and then eventually with Fabian Theis about how can we think about what are the particular properties of these challenges in machine learning that make them really amenable to that computer science audience, and how can we start to translate the problems that we're facing in single cell biology into that framework. And so what we really arrived on was this concept of open problems. And so in machine learning, open problems are really one of the bases for driving innovation in that field, or in those fields, we should say. So on the left, I'm showing here a screenshot from Papers with Code, which is a product of Facebook AI research. They house or host a set of state-of-the-art leaderboards for a number of different kinds of tasks. And when you look through these, you realize that there's 5 key properties of open problems. So the first is that there are formally defined tasks. And what I mean by that is that there are problems that are well posed, that you can understand whether or not a solution is actually approaching a particular task. It's unambiguous. There are also easily accessible data sets. So if you want to do, say, image classification, there are a set of standard data sets that almost any new algorithm gets benchmarked against, and these have reliable ground truth. There are also a set of generally agreed upon quantitative metrics that you can use to evaluate a given algorithm and determine how well it performs at a given task. And then there are many benchmark methods that are all sort of compared against the same baseline in these leaderboards that are often continuously updated over time. And so this was, I think, really an exciting framework for us to start to think about how to formalize challenges and to drive an innovation in single-cell analysis. Another interesting thing that we learned as we're starting to dive more into this space of machine learning is that there's a really crucial value to doing benchmarking in a centralized way. So this is a figure from a review last year of benchmarks in a field called metric learning, of which dimensionality reduction is a subset, representation learning is kind of a general analog. And on the left, what we can see are benchmark metrics on 3 standard tasks over time, ordered by different papers. And if you just look at the average performance on these benchmarks over the past couple of decades, 15 years or so, what you see is that it looks like it's increasing on average. In this review, what they did was actually take all of the algorithms presented in these papers, or in at least a subset of them, and run them on the same benchmark methods, but make sure that the data sets were all processed in the same way, that all of the algorithms were using the same set of parameters when they were being compared against others. And what they found is that when you do benchmarking centralized, you find that maybe some of the metrics can be different than they might be when they're done by someone developing a new algorithm who wants to show the improvements that their algorithm generates. And so we love this idea of open problems and having them be evaluated in a centralized manner. And so over the following year, over the year of 2020, a bunch of folks in Smitha's lab and folks at CZI Biohub and at Helmholtz in Munich came together, and we created this open problems framework. So there's a website you can go to right now, openproblems.bio. And there's also a GitHub repository, openproblems-bio, that houses all of the code that we're using to organize these centralized benchmarks and formalized tasks in single-cell biology. And I'm not going to get too much into the weeds of the technical details of how these repositories are set up, but I just want to highlight a couple of aspects. We're running repositories that both load data, contain many different sets of tasks that are community contributed and are evaluated on the cloud. We're using some exciting practices to make sure that code can be contributed in multiple languages and all evaluated on the cloud in an automated manner. And this has been, I think, a really exciting way for us to actually build infrastructure for these centralized benchmarks. I was also really excited that in March of this year, CZI helped us bring together 60 different scientists from 34 universities around the globe. We actually had even more people than who registered join us on our Discord server over 3 days to contribute new methods and new tasks to open problems. We played a lot of single-cell Scribble, which is sort of like a version of Pictionary you can play over Zoom to bring together folks in an informal setting that's kind of difficult to do when you're hosting a virtual event like this. And we had a lot of really meaningful contributions to our GitHub repository. So there's 70 new issues, or code contributions that people wanted to contribute, and we had 11 new pull requests, more than half of which actually are already merged into the main code base. And just to give you a sense of like the organization that the community brought to this is just one task board for the dimensionality reduction task. And a bunch of folks came together to sort of lay out how they wanted to frame and formalize this sort of idea of reducing dimensionality of a data set for visualization and to something that you could quantify and benchmark in a formal way. What was also really exciting at the end of that workshop is that the organizers of Open Problems, along with some other of our scientific collaborators, submitted a competition to NeurIPS for 2021, that conference I mentioned at the beginning. They host a set of machine learning competitions every year, and ours was one of 23 to be selected for this year, and the only one in the life sciences. This is like one of a handful of life sciences competitions ever to happen at NeurIPS, and I think the first one in single-cell biology. So the title of this competition is Multimodal Single-Cell Data Integration, and this is most of what I'm going to talk about for the rest of the talk. So as we came to think about how we wanted to take this idea of open problems in single-cell analysis and introduce it to the machine learning community, one of the things we realized we needed to think very deeply about is how we were going to introduce the concept of ground truth, which is difficult to get in biology, but necessary to involve the machine learning community and really important for establishing formalized benchmarks. And when you look in machine learning, I like to think of there being three different kinds of classes of ground truth. So one of them is expert annotation. And this is really common for tasks in image classification. So you can see ImageNet, which is a collection of 4 million images that humans have gone through. So every image has like 3 or 5 people look through and actually say, this is where the fox is in this image, and that there are 3 of them. They do this manually, right? This is something you can look at images of digits and say, that's a 4, that's a 2, that's a 1 with very high confidence, then train an algorithm to go and actually label them in an automated manner. This is tricky to do in single-cell biology because not only is the whole concept of developing cell ontologies to describe cell state field of active research, it's also a very high barrier. You can't just take anyone and have them annotate an individual cell profile at an expert level like you can with an image. You need to have typically years of experience in the field, and so there's a much smaller pool of people. It's much more expensive to get expert annotation for single cell data. The second class of algorithms in ground truth is sort of measurements of dynamical systems. So this is when you have the same thing that you're measuring repeatedly over time, and you can develop tasks around trying to predict future behavior or outcomes of those systems. We see this a little bit in life sciences, especially in the field of electronic health records. So the MIMIC-3 data set of ICU admissions measures patients over time along with their outcomes. We also see this in weather forecasting. This is difficult to do again for single cell biology because we don't really have omic methods, measures of measuring like the whole transcriptome repeatedly over time to give a holistic temporal view of what a cell is doing. Finally, there's this field of multimodal measurement. And if you think about this abstractly, machine translation, so WMT stands for the Workshop on Machine Translation. This is a kind of multimodal representation of the same sentiment. We can have a sentence in Russian and then ask it to be translated into English, or speech to text. Again, we have 2 different modes of representing the same idea, and the goal is to train an algorithm to go back and forth. And one of the things that's really exciting about multimodal data is that we're now at a time in 2021 when we've been starting to see multimodal single-cell omic technologies developed at scale. So in 2017, we had the first RNA and protein measurements. In 2018, we had some of the first RNA and ATAC-seq measurements in the same cell. And then in the following 2 years, commercial kits became available that allow us to do this in a robust and reliable manner. So what does it look like to measure RNA and ATAC-seq in the same cell? So you can imagine we have this representation of an individual cell. What you can do is isolate the nucleus from those cells and then measure the accessibility of chromatin in the nucleus along with nuclear located mRNA and get across the entire genome a measure of how accessible the DNA is. And then also for every gene, a measure of how highly expressed it is. And you can do this for tens of thousands of cells. Similarly, you can measure RNA and protein in the same cell. Now, this is not quite an omic technology because we can only measure right now about 150 proteins in an individual cell. But what we can do is measure all of the 20 to 30,000 genes, both in the cytoplasm and in the nucleus, along with 150 different markers on the surface of these cells, and then link together in individual cells, both their expression and their abundance, and try to understand the relationship between those two. So with these technologies set up, our next question was, what are we going to actually look at? How do we want to measure these as we're trying to design a benchmarking data set? And we aligned across a number of our partners on bone marrow mononuclear cells, or BMMCs. And there's a few properties that make BMMCs really attractive for doing multimodal analysis. So one is that the system is very well understood. We know the different kinds of cells that are present in the bone marrow. We also know what surface markers are expressed on them. As you can see on the right here, this is 100 cells from the Human Cell Atlas. And there's not just one sort of cluster structure or trajectory structure. There's actually a mixture where there are, definitely differences between, say, T cells and B cells or T cells and natural killer cells, but there's not necessarily, but there are also shades of gray within those compartments. So there are, spectrums, and there are clusters in the data set that make this a really exciting arena to do machine learning in. It's also relevant to human health, which is something that CZI and Celerity and the genetics department at Yale and Helmholtz and BioHub are all really excited about. And then finally, they're commercially available. You can go to a vendor and ship these cells all around the world from a diverse set of donors without needing to have special licenses or relationships in place. And it helps that a lot of the surface antibody panels are focused on immune cells. So with the technology and the system set in place, we came together with our 4 partners and designed a fit-for-purpose benchmarking data set for multimodal data integration. And the way that we decided to set this up is that across 4 sites, we were going to take 9 donors and purchase bone marrow mononuclear cells from them, and then split up the measurement both of ATAC-seq and RNA and proteins across each of these locations. And so donor 1 is going to be measured at all 4 sites, and then each site will have 2 donors that are measured uniquely at those sites, both in the ATAC and RNA and protein level. And then we're going to split it up. This is just one proposed split. We're actually going to shuffle, which is the training and test. But the idea is that during the training phase, individual competitors will have access to samples from 7 donors from 3 sites, and then we'll use a remaining site for testing when the competition ends in the fall. BioHub was really excited about getting involved with this, and so they actually also are generating multi-omic data from liver, kidney, muscle, and lung of 4 different human donors. And so there's going to be 4 samples of multi-omic data, and this will be really helpful for us to understand how do algorithms trained on bone marrow cells apply to other tissues within the human body. So now that we've outlined the data set and the context for this competition, I'm excited to describe to you the different tasks. And I think one important thing to note is that each of these tasks in this competition come with a $5,000 prize that Celerity is contributing for the algorithms that perform the best in that category. The first task is predicting RNA from ATAC at single-cell resolution, or also protein from RNA. So this is modality prediction. A basic overview of this kind of task is you can imagine having for a set of cells, the ATAC-seq profiles, the chromatin accessibility, and then the task is to predict in each of those cells the corresponding expression measurement. We can then rank an algorithm's performance using a metric like mean squared error and say how well do the true values correspond to the predicted values, and the closer you are, the better you perform. The second task is around learning integrated representations of multimodal data. And so again, I'm showing you ATAC-seq and RNA-seq, but we can also do this for RNA and protein. And the goal is to take multimodal measurements across cells from each of the donors, and then to learn an integrated representation of the data in a reduced dimensionality that preserves meaningful biology. And so the kinds of things we might measure here to score performance are actually quite similar to how you might think about this challenge in batch integration. So you can look at alignment in this reduced dimensionality, and it would be bad if you have all of your ATAC-seq profiles on one side and all of your RNA-seq profiles on the other. It's good if you have the cells more mixed, and there are ways you can quantify how well mixed the 2 modalities are. We can also think about trying to preserve the cell cycle across different populations of cells within the data set such that we observe the expected mixture as opposed to just seeing clumping that has to do with the way that these markers are identified in different kinds of cells. Finally, we can think about looking back towards this idea of expert annotation, which is to some extent possible in immune cells, and see how well do similar cell types group together in this dimensionality reduction, and how well separated are different ones versus just a lack of structure of known immune compartments in your embedding. And so you can look at this across many different sets of metrics and come to an average performance to rank algorithms. Finally, the third category is a problem of matching 2 modalities. And so not every data set actually measures both of these in the same cell. And for other reasons, we're actually interested in knowing if I give you, for example, an RNA profile, and I don't tell you which ATAC profile it's matched to, how well can you match the correct other modality. And so here I'm showing RNA to attack prediction. And the idea is that for some RNA profile 1, which is just a shuffle representation, of the data, you might say, well, I have a certainty of 0.4 that this matches attack profile 1 and a certainty of 0.2. I'm like less confident that it's this second profile, and I'm about equally confident it's the third. And what you can do is try to do this matching across all of the profiles, such that the predictions sum to 1. We can then look at the ground truth because we actually know which profile matches to which, and then you can generate scores based off of, where the lowest possible score is 0, you got all your predictions wrong, and the highest possible score is the number of cells, where you have 100% certainty that this profile matches that one. So here, because this guess isn't so confident, you only get a score of 1.7 as opposed to the total score of 3 available in this small setup. And so those are sort of 3 tasks that we're introducing for this competition. I want to make sure that I recognize that this is a very deeply collaborative effort with a really large number of both academic and industry partners. It's been really wonderful that Celerity has been contributing to data generation and also is offering prize money for this competition and allowing me also to continue to organize this work as a scientist there. I'm also incredibly appreciative for the Chan Zuckerberg Initiative, who is helping to pay for data generation across all of our academic partner sites, and also paying to help support the computational infrastructure for the project. I'm very appreciative to our partners at BioHub, Yale, and Helmholtz, who are helping to generate data and also offering financial support in terms of reagents and labor. I'm also really excited that we have a few other industry partners that have just gotten interested in what we're doing and offered to get involved. Saturn Cloud is going to help us make this competition much more accessible. I'll talk about in a second. Data Intuitive is a consultancy that's helping us to put together the computational infrastructure. BioLegend creates those antibody surface marker panels. They actually offered us pre-market the version 2.0 panels for this system for free, which was really exciting. And then we have a number of academic partners at Harvard, Mila, and ETH Zurich who are helping organize the competition. As I sort of mentioned, what Saturn Cloud is doing, this is, I think, going to be really important for folks to try to get them more interested and involved in the competition. They're actually going to produce a dashboard like this. So we use Saturn Cloud internally for some of our cloud compute, and you can just click a button and launch an instance that will load data for this competition and then allow you to start doing compute for free on servers that they're going to pay for, something like 100 hours from up to 1,000 competitors. It's really exciting. And finally, I want to encourage anyone here who is interested in what I've been talking about to go to openproblems.bio/NeurIPS2021, hit this sign up button. There'll be a form you can fill out to give us some information about yourself. And then as soon as the data is up and the competition is started, we'll make sure to keep you notified. I also would encourage everyone to go join our Discord. You can go to this shortened URL here, introduce yourself, say hi, start to think about who you might want to form teams with. And finally, I just want to encourage people who maybe are interested but not sure if they want to compete themselves. Please do hit the sign up button and at least give us your email address, because we're going to send out some communication, maybe a video, describing this competition over the coming months. And we really want to get as many different people from machine learning, from computational biology and bioinformatics and computer science interested and involved in this competition. And if you can help get the word out to your network, we'd be incredibly appreciative. So with that, I'm going to sort of end the talk here, and I'm happy to take any questions.